Hi Komal, welcome to Quantum Photonics.
Hi, Cecil. Thank you for having me.
Yeah, thank you so much. So we have up here our co-moderator who is on stage who also wants to welcome you. So yeah, we will be starting shortly. In the meantime, we will be waiting for more people to come in. Yeah, let's go for it.
And with that, the mic is yours.
Welcome to Quantum Photonics. So we are very glad to have our guest here, Komal Bhattacharya, who will be presenting her paper. And I would like to inform the audience that I had posted the link of her research and also the popular article in my Twitter account, and I had posted it as well in the room chat here. So please feel free to refer to these references. So yeah, I would like to introduce our audience for today. Komal Bhattacharya from Max Planck Institute did her bachelor's and master's in India in the Indian Institute of Science, Education and Research in Kolkata in physics with a focus on theoretical biophysics. Then she joined as a PhD student in the group of Professor Dr. Karen Alim in the Max Planck Institute for Dynamics and Self-Organization in Göttingen, Germany, almost 4 years ago. In this group, they do research on biophysical properties of flow network with a focus on the organism Physarum polycephalum or slime mold. During her PhD, she worked on theoretical understanding of memory in flow network morphology using numerical and theoretical tools. So yeah, I'm very proud to present our guest, Komal Bhattacharya. Komal, welcome to Quantum Photonics.
Hi, Cecile, and hello, everyone. And thank you, Cecile, for this amazing introduction and for having me in this group. So, as Cecile introduced, I'm a PhD student working on theoretical understanding of memory formation in adaptive networks. And today I'm going to talk about a very recent publication from us about how memory is formed in adaptive networks. If you go into my slides that I created, if you go to the first slide or the second slide, let's say, I introduced like what adaptive networks are. Adaptive networks are part of our lives, starting from the transport network that we use every day to go to work, to the social network that we are part of. All of this adapts all the time following variety of conditions. Our relationships to our peers, to our friends, to our colleagues change all the time, which is basically the connections of the social adaptive networks. Similarly, another network which is very much part of us is our blood vasculature network. And this blood vasculature network also adapts all the time depending on variety of conditions and primarily depending on the amount of blood flow going through these networks. Blood vasculature networks are flow networks. And in this work that we presented in this particular publication, we primarily focused on this kind of adaptive networks, which are adaptive flow networks. But a lot of the results that I will talk about here are also usable in other kind of adaptive networks as well, not necessarily just flow networks, but mechanical networks or resistor networks. If you go to the third slide, the next slide, I introduced the different kind of flow networks that are observed all over the nature. Because flow networks are very common and very abundant in nature. They're not just part of our blood veins, they're also seen in plant as water vasculatures. They're of course seen in animals as blood vasculatures, the blood veins, and they're also seen in organisms like Physarum polycephalum, which is an amoeboid organism, which is a single cellular organism but with multiple nuclei, and it grows in shape of a flow network. And this organism has shown a variety of very interesting and very, very intriguing behavioral property, although it's only a single cell. And it has shown a lot of very intelligent behavioral properties as well. If you go to the next slide, I have included a link to a video with this slide, which shows how this organism is moving over the time. So this organism, which is a single cellular organism with multiple nuclei, is shaped, it's a single cell, which can be huge, and it grows in shape of this network, and this network constantly changes and adapts and moves around. And this organism is what is called slime mold that our group is working on. And this video shows a snippet of this slime mold moving through a petri dish. And at minute 0, a food droplet is offered to this organism, and this organism moves towards the food droplet. Maybe you can go to the next slide, where I have actually focused more on the snapshots of the video itself, which is interesting to look at, which shows how this organism, which is a network, is moving around and changing its shape, the network shape. At minute 0, a food droplet is applied to the network. If you can see it in the snapshot of the video named minute 0 in this slide number 5, at the location which is indicated by the red arrow. And when the organism senses the food, it basically changes its migration direction, because before it was migrating from left to right. If you go back to the video, you can see that the organism was moving from left to right. And when the food is applied, it changes its migration direction and it starts moving towards the food. You can see it at minute 90 that the organism basically reaches the food at minute 90 in the snapshot you can see. And what happens after a long time at minute 310 is that the organism completely consumes the food. But even though the organism has completely consumed the food, the organism still retains some big tubes around the location where the food was applied to. If you follow the minute 0 snapshot again, where it was shown where the food is exactly being applied to, and you come back to minute 310 snapshot of the video in the same slide, you can see that there are these big tubes which are formed around the location where the food was applied to, even though the food is completely consumed. And these big tubes are retained in the network for a very long time, even after the food was consumed. It's kind of a way for this organism to remember the information or the stimulus inside the network morphology. And because this organism shows an array of intelligent behavior, this property of how this organism is creating this memory of a stimulus in the network morphology itself without any neural networks is very interesting for scientists to study. But this memory formation, or this, this imprint of a stimulus into the network morphology is not something which is unique to just the network of this organism, which is basically a flow network, but this is also seen in our blood vasculature as well. If you go to slide 6, I have created a schematic which shows the after effect of a stroke in the brain microvasculature. What happens is that in brain microvasculature, if a blood clot blocks the cerebral artery, which creates an ischemic stroke in the brain microvasculature by blocking the blood flow in a certain region which is downstream from the artery, what happens is that even after opening, or even after by using some kind of medical procedure, removing the blood clot, even after removing the blood clot by using some medical procedure, the blood flow in this downstream region, which I have shown in the schematic by this pink shaded region, does not go back to, the blood flow in this pink shaded region does not go back to normal. The blood clot is shown by the yellow and black region in the middle schematic, where the blood clot is actually directly blocking the blood flow, through this pink shaded region, and the last schematic shows that even after removal of the blood clot, the blood flow in this pink shaded region does not go back to normal. If you go to slide 7, where I have shown actually some actual sample of the brain microvasculature, which shows this particular property, where one can see is that in the left slide of the microvasculature, which is the control picture of the brain microvasculature, the network or the vasculature is completely filled with blood. But after, as the right plot shows, after 15 minutes of bloodless ischemia, if the blood clot is removed and the blood flow is allowed to go back to normal after the stroke, if the blood flow is allowed to go back to normal in this brain microvasculature, what we see actually that the blood flow actually does not go back to normal. That means the brain microvasculature somehow keeps the imprint of the stroke in the network morphology. That means it is also keeping some kind of memory of the flow in the network morphology itself. And this is what we want to study in our work, that how is this kind of memory is formed in flow network in general, starting from the flow network of Physarum polycephalum, this organism that I showed you before, to the flow network of blood microvasculature. Now, how can one define memory? Because the concept of memory is, of course, not so easy to understand, and especially in the context of something which is not human and not an intelligent animal. How can we define memory in such a system? What we see is that we can say a system has a complex system. Not necessarily our brain is also a complex system, but not necessarily our brain neural network. But in general, any complex system can be considered to have a memory if there is any process of writing an information or imprinting an information of a stimulus into the system, and there is a process through which the information about the stimulus can be read out from the system after a certain time. And we can consider this information as memorized information only when the output read out from the system is correlated to the information that was actually written into the system. So now we are thinking about memory from a very, very general perspective, not just thinking about the memory we have as human, which is also fits into the same definition of memory, where we read something or we get some kind of information or get some kind of knowledge or skill in our system, in our brain, in our neural network, and we use that, we read out this information much later, even though when we are reading out this information is actually much later after we have actually written the information in our neural network. And we can consider this information to be memorized information only if the information, the output that we are reading out, that we are using much later, is correlated to the input, and only if they're temporarily separable in general a complex system. And if you through this talk, basically I have created an outline in slide 9 where I am explaining basically what I will describe during this talk, during this presentation. In slide 10, where I show that the first part, what I will talk about in this talk is basically the model of biological flow networks that I'm going to use, because I'm going to study the memory formation in this kind of adaptive flow networks. If you go to slide 11, here I have shown how I am modeling the biological flow networks. So, I model the biological flow networks as networks made with hollow cylindrical tubes which are straight. So I consider the network is made with many different hollow cylindrical straight tubes. And if I consider each of these links of these networks as hollow cylindrical tubes with fluid flowing through them, one can use this Hagen-Poiseuille equation, which is a very simple linear equation, to write down how the flow through these tubes are going to be as a function of the pressure differences along the tube, which is a very simple linear equation similar to the relationship of current to voltage drop following Ohm's law. And the cofactor that comes in, which is the proportionality constant, which is the conductance in case of current network, also they're existing in case of fluid flow network. And in case of fluid flow network, the conductance is basically conductances of, or how easy or difficult it is for the fluid to flow through this one tube depends on the radius of the tubes and the length of the tube, as well as the viscosity of the fluid. In the next slide, I show that the flow network can be considered as network made with one of these hollow cylindrical tubes attached with each other. And so, following
that is slide 12, right? For the sake of those who just joined the room.
Yes, I am currently talking about slide 12, yes. Yes, I'm talking about slide 12 where I'm showing how the flow network can be modeled as a very simple network made with hollow cylindrical tubes where the flow is directly proportional to, linearly proportional to the pressure difference through these tubes, which makes the network which allows us to fit the network, the flow network, to a current network, because the pressure differences can be considered can be mapped to the voltage differences through current networks, where the flow through the tubes can be mapped to the currents through a current network. And we can also consider, similar to current network, that the Kirchhoff's law of current is valid in case of these flow networks, which basically means that the total flow through one node has to be conserved, has to be zero basically. Whatever flows in, flows out. So following this very simple idea, very simple linear system, we model the flow networks in our work. Now if you go to slide 13, where I discuss about how the biological flow networks, the adaptation of biological flow networks are considered. Because as I'm going to be talking about the memory formation in adaptive network, I want to know how the adaptation actually occurs in these biological flow networks. So, the main purpose of biological flow networks in general, as example, our blood vasculature system, let's say, or the vasculature system in the water vasculature system in plant, or the Physarum polycephalum, the organism that I talked about. In general, the main purpose of biological flow networks is to transport fluid from one part of the organism to another. And through the fluid, these biological flow networks transport all kinds of nutrients and resources and hormones, like in case of plants and us, the hormones and nutrients and oxygen throughout our whole body. So this main purpose of these biological flow networks in general is transport. So there has been a theory since 1926 it has been established since 1926 by Murray, that these kind of biological flow networks actually adapt to minimize or maximize transport by minimizing their energy dissipation, because the main purpose of these networks are transport. And here I show basically through the builds of this slide in slide 13 how that we consider that these biological flow networks minimize their energy dissipation, which is shown by the first build into the slide. The second build into the slide shows that, but this biological, the amount of material to create these networks are conserved, because we need resources, we need material to create this network. If we want to make a new blood vessel, we need cells for that. And so to minimize, along with minimization of energy dissipation, which maximizes transport through this network, we also need to maintain the available material into this network. And if we follow this minimization of energy dissipation and the constraint of available material, we can come up with a very simple solution, which is shown by the third build in the slide 13, a very simple equation which shows how the conductances of the networks that I talked about in the slide before, they adapt over time. The term in the left-hand side, shown by the fourth build, talks about how the conductances of each of the links or each of the tubes in this biological flow network that I talked about, they adapt over time. And the terms in the right-hand side, the term in the up, shown by the fifth build, talks about how the tube conductances update as a function of the flow through the same tube. And where the term in the bottom, shown by, in the right-hand side, shown by the sixth build, talks about or incorporates how the tube conductances update inversely proportional to the flow through the whole network. Basically, keeping in consideration of minimization of energy dissipation as well as the constraint of the available material to create such network, we formulate this adaptation rule, which makes sure that the conductances of the network changes or adapts to flow for increasing the efficiency of the network. Okay, this was a lot of equations in this slide. So maybe if we go to slides 14, maybe I can try to simplify the whole idea to you. So the idea shown by the slide 14 is that what these networks are trying to do is basically they are trying to optimize the transport through this network, because that's the main purpose. So what that means, if there is a network with 3 tubes as shown by the plot, shown by the schematic in this slide, in slide 14, if the flow through 2 of the tubes is higher than flow through 1 of the tube, to maximize the transport of flow through this network, what the adaptation dynamics will do is basically they will increase the radius of the 2 tubes which has higher amount of flow, shown by the third picture, shown by the middle picture in the schematic. But as the amount of material to create these tubes are constant, what that will do is basically that will reduce the width of the tube or the radius of the tube with very less flow to provide for the material to grow the tubes that has high amount of flow. And this is basically what our adaptation rule that I just showed in slide 13 is basically incorporating. The maximization of transport by changing the conductances of the tubes, by changing the radiuses of the tubes to flow, and maintaining the amount of material simultaneously. If you go to slide 14, If you go to slide 14, here I show an example network that is created by us as an initial condition. So we basically create a network, a flow network, using this model of flow network that I just described before, which has a sink in the center of the network, and it has sources of inflows at every one of the nodes in the network. That means there are external inflows that are coming in this network that I have shown here. And what I do is I apply an extra inflow as a stimulus to the network, which will be shown by the green dots. And if you go to the second build of the slide, you can see how the network will adapt if I follow the model or the adaptation rule that I just described before, that the tubes with high amount of flow will grow and the tubes with low amount of flow will shrink, maintaining the available material. The slide 15, second build of the slide 15 shows how if we let the network adapt over time following this kind of adaptation rule, how they are going to look after a certain amount of time. And here the green dots basically signify an extra amount of inflow that is coming into the network through these nodes. And this is what I consider as a stimulus to my model of adaptive flow networks. And we see that there are these big tubes which are connecting the stimulus, the green dots, to the center of the network, to the red dot, which is basically the sink in the network. So from the source, the flow is flowing through the whole network and going through the sink of the network out, because the total amount of flow has to be conserved in the whole network following the Kirchhoff's law. So after establishing the model of biological flow networks, if you go to slide 16, I start now thinking about how to quantify memory in such adaptive flow networks. And as you can understand, it's not so straightforward to quantify and understand memory in adaptive flow networks. In slide 19, through the builds of the slide 19, I show how I attempt to read out the memory of an extra local inflow shown by the green dots, as I described in a few slides ago. How we can read out the memory of this local inflow or stimulus in an adaptive network. So I start again from an initial network which has no particular polarity to it, so it's filled with tubes with random conductances. And I apply an amount and a local influx or a stimulus to this network at an angle θ1, shown by the first build of the slide, as you can see shown by the arrows towards the green dot. So here is where I apply the stimulus in form of a local extra inflow to the network. And if I adapt the network with this inflow for some time, if you go to the second build of the network, you can see that how the network grows into after a certain amount of time. It grows these big tubes connecting the center to the stimulus location, which is the additional inflow. And now if I take this additional inflow off from the network, or the stimulus out of the network, to quantify if the network can still remember the information, what I do is basically that I take the stimulus off and I just adapt the network without any additional stimulus for some time, which is shown by the next build into the slide. And you can see into the next build that the network grows into this kind of homogeneous looking network without any particular polarity. And now we ask the question that if this relaxed network, so I first train the network with the stimulus and then I take the stimulus off and relax it for some time, And then I ask the question if this relaxed stimulus still remembers where the stimulus was applied to. And to probe that, what I do is basically I apply a second stimulus, a probe stimulus, to the network shown by the next build in a different angle θ2 shown by the next build to check how the network reacts to this probe stimulus if I change the direction of the probe stimulus shown by the next build. So that means here we have three sections in our protocol to read out memory, which is shown in slide 18. And if you go to slide 19, here I show how the three different sections come together as the memory definition, which I talked about before. So the first section is basically where I write the memory, write the information in the network by training the network or adapting the network with this additional inflow, or which is a stimulus. I train the network with the stimulus for some time, which is the writing process. Then I relax the network for some time, which is basically the temporal separation between writing and reading, which is necessary to understand if something actually has memory or not, with some noise. And in this part, basically, I don't have a stimulus, and I adapt the network with just fluctuating inflow at all the nodes of the network. And in the third part, what I do is basically I apply another stimulus, which is a probe stimulus, to read out the information written into the network. And as you can see in the plot of the output, as an output of the readout, what we plot is basically how the network reacts to the stimulus that is applied to the network, and to the direction of the stimulus that is applied to the network, to the probe stimulus. And we measure the energy dissipation of the network along with the direction of the probe stimulus. And we observe that whenever the direction of the probe stimulus is exactly same as the direction of the, this is shown by the last build in the slide 19, we observe that whenever the direction of the probe stimulus is exactly same as the direction of the writing stimulus, the energy dissipation drops. That the energy dissipation is significantly different for the trained network than for the untrained network. That means somehow the network remembers where the input stimulus was applied to. It remembers the direction of the input stimulus, and when we are looking at the reaction of the network to different probe stimulus, it reacts significantly different whenever the probe stimulus is applied exactly at the same direction as the input stimulus. That means even after relaxing the network with noise, it remembers where the stimulus was applied to. It has a memory. Now if you go to slide 20, we talked about the model of biological flow networks and how we quantify memory in such network. And in the next part, we describe how the network hierarchy is storing the memory of a stimulus in the network. For that, what we did was that we measured the dynamics of the conductances, of the link conductances, with respect to the conductances itself in the network. What we observed in our work is that the conductances which are smaller than a threshold shown by the vertical dotted pink line continues to decay over time, where the conductances which are above this particular threshold is more or less constant. This is shown by the change of conductances from one iteration to other, because in the y-axis we are plotting the conductances in the next iteration with respect to the conductances in the current iteration. So if the y value is actually smaller than 1, that means the conductances in the next iteration is going to be smaller than the conductances in the current iteration. That means they will decay. And if they're greater than 1, that means the conductances will grow in the next iteration. And what we see is that if the conductance values are below this particular threshold, that this ratio of conductances are always less than 1. That means the conductances below a particular threshold always decay. And not only that, but we also see that they follow this power law relationship when plot power law relationship to the conductances itself. That means the smaller the conductances are, the stronger the decay of the conductances are. That means if a conductance starts decaying, if a tube starts decaying, it will just continue decaying. There is no way of growing it back. And we see that this dynamics is actually very, it's observed for all kind of parameters. So even if we change the parameters, which is shown by the next build in the slides 21, what we see is basically that the conductances show the exact same dynamics for a different set of parameters. Only shown by the gray dots, you can see. Only the threshold value below which all the conductances decay, that changes depending on different parameters. Okay, maybe this plot was a bit complicated. This plot, what it means basically, which is shown in slide 22, is that the networks which is shown by a network which is shown by the first build into the slide, where all the thin tubes are colored by blue and all the thick tubes are colored by red. What we can see is that all the thin tubes, which are colored by blue, will end up decaying after a certain amount of time. If you go into the next build, into the slide 22, you can see a network which grows over after a certain amount of time, has none of these blue tubes, and it only has tubes which are of the color red. That means the very thick tubes are the only tubes that are going to retain in the network morphology after a certain time. And this hierarchy that is being established is irreversible. There is no way of growing a tube back after a time. And this irreversible dynamics of these tubes, this plasticity, is what is responsible for storing memory in the network morphology. And if we go to slide 23, we show in our work, in our publication, that we can even predict this dynamics that is controlling the memory formation mechanism in these adaptive networks, even by using some very simple analytical calculations. In slide 24, I show what is the formulation of the analytical calculation that I used. So for analytical calculation, we consider a very simple network with 3 links. And now the one link of the network in my calculation does have very small conductance values, which is shown by the red dotted link. And the blue links have stronger conductance values. In this minimal network setting that I used for analytical calculation, the node 1, shown by the red dot, is basically a sink in the network, and the node 2 and 3 has inflows coming into it. That means that means that all the inflows that are coming in the link 2 and 3 will flow through this minimal network and go into the sink node of the network and flow out of the sink node, which is defined by the node 1. And what happens is basically so now if we consider that the link between the node 2 and 3 is very small, then what will happen is that all the fluid that is going through into node 2 and node 3 will go directly to the sink at node 1, and then there will be no flow through the link between 2 and 3. So now what we do, and this is at a certain time point. And now if we consider in the next time iteration shown by the second build in the slide 24, in the next iteration what we consider that we add a perturbation in the inflows in the network at the node 2 defined by delta Q. And now we want to see that if we add a perturbation in this very simple minimal network, how the network conductances react to this perturbation. Do they keep the same values that they had before? Do they continue shrinking? Do they grow? How do they react to this perturbation, to this minimal network? Is what we check in our analytical calculation. And when we try to check how the conductances will change as a reaction to this perturbation applied to node 2, what we find shown in the slide 25 is the dynamics, exact dynamics that we observed for the conductances from our numerical simulations of a very complicated big network. What we observe is in this case as well that the link which had very thin values, which had very small conductance values, that means the link which was very thin, the connection which was very thin, which is the red dotted line. Basically in our minimal network shown in slide 25, what we see is that the conductance values follow the same dynamics of the conductances from the numerical simulations below the threshold. That means they follow the same dynamics of the small conductances which we also observed in our numerical simulation. And the dynamics of the conductances of link 1, 3 and 1, 2, which are shown by the blue colors in our minimal network, they follow the same dynamics as the big conductances found from our numerical simulation. And similarly, we could also find the threshold value below which basically the conductances behave differently, also from this very simple analytical calculation as well, which matches with our numerical simulation. What this all means is that the analytical prediction showing the analytical prediction using this very simple minimal network showing the same dynamics of link conductances of the irreversible dynamics, showing this irreversible dynamics also tells us that basically the adaptive dynamics adaptation dynamics itself is which is leading to the memory formation in the network morphology. That means we don't have to include much more complicated complications into the model to get memory formation in the flow networks, because even the simple adaptation dynamics itself is leading to memory formation itself. Till now we talked about flow networks made with hollow cylindrical tubes. Now, to check if our hypothesis that the adaptation dynamics itself is leading to the hierarchy change or leading to this irreversible dynamics of link conductances, which is leading to memory formation, which is our hypothesis, if this hypothesis is correct or not, we want to try out different kind of networks which follows different kind of adaptation rules. Okay, so maybe I will give you a bit of introduction to what I'm talking about. So if you go to slide 27, so till now, the first picture shows you in the left shows a hollow cylindrical tube, and this hollow cylindrical tube has a particular form of material conservation where this conductance to the power half term pops up in the equation of material conservation. But if we use a different kind of network, as example, networks made with porous tubes, shown by the first build in the slide 27, what we see is that how the amount of material needed or how the conductances relate to the available material changes, which also changes how much we need to move material from one tube to another to basically grow a tube or increase the efficiency of the network. And we can consider a general constraint of available material of networks, of adaptive networks in general, by assuming this parameter gamma, which is shown by the third build into the slide 27. So basically what this gamma factor determines is basically how the conductances of the network is related to the available material of the network. As I talked before, that the available material really is important for the network adaptation, because the available material determines if a small tube needs to shrink to allow for the growth of another tube or not. So how the conductances are related to the available material or the mass or the material itself will determine how much a tube would have to shrink to allow for the growth of other tube to increase the transport efficiency through the network. So this constraint of available material is actually very, very crucial. And this constraint of available material also changes how the network structure is going to look like. So if you go to slide 28, I show in the first build, in the first picture in the slide 28, that the networks which are made with hollow cylindrical tubes, they have this hierarchical tree-like network structures with a lot of very big networks and some very small branches coming out of these big branches, like tree-like networks basically. But if I make a network which is made with porous tubes, shown by the second build or the next build into the slide 28, if we consider the network is made with this kind of porous tube, which is actually observed in case of plant vasculature system, what we see is that the network which is created, or this adaptive network that is created, or the stable network that is created from such tubes, is basically a very loopy network. It has some hierarchy in the network, but it also has all these small tubes intact in the network, which are shown by the green colored tubes in the network. That means there are no tubes which are decaying irreversibly in this kind of network, which is also shown by our analytical calculation as well, that the tubes that we can also calculate it analytically as well, how the conductances for the small tubes are going to change for different gamma, and we see that for gamma equal to 1, that the change of conductances for the small tubes is supposed to be equal to 1. That means they are just supposed to stay constant and not decay. And when we plot the conductances, how the conductances adapt over time for conductance values itself, for gamma equal to 1, we see that this is in fact true, that the conductances do not decay irreversibly over time. And as we know, for hollow cylindrical tubes decay irreversibly over time and retains memory. We also assume that the network which are not made with hollow cylindrical tube and made with porous tube this kind of network will not have memory. And in slide 30, in the first build, we show that this is in fact true. And when we use the method to read out the information from the network morphology by probing the network from different direction and looking into the reaction of the network to see if the network still reacts differently when probed from a certain direction or not, what we see is that there is no significant difference between the reaction of the network given by the energy dissipation of the network as a reaction to the probe stimulus, which can, there is no significant difference which can be read out as memory. That means that this kind of network, which is very loopy and does not have a strong hierarchy, cannot retain the memory of an external stimulus in the network morphology. To summarize in slide 31, basically we observe in the first point is that the memory effect is observed in flow networks. As the second point, we observe that the irreversible hierarchy created by this decaying weak link is responsible for the memory effect. And as the third point, we observe that these irreversible dynamics are originating actually from the adaptation rule itself. And as the fourth point, we observe that the memory is not conserved in networks with low hierarchy. As an outlook, we can, shown by the slide 32, we can look into different effects of memory, like how memory effect is changing as a function of different system parameters like the strength of the stimulus, the inflow fluctuation, the conductance fluctuation, the training time or the relaxation time of the network. And also we can look into how memory capacity of adaptive network change, which is a work which we have very recently put into archive. You can take a look at it if you like. And thank you for listening. And I thank my supervisor and our collaborator, Dr. David Zwicker, and my supervisor, Professor Karen Alim, for this idea and helping me with this project. And I thank you all for listening. And I'm up for any questions you might have, anything that was not clear in my discussion, anything that you might have, any ideas, anything you have come up with. Please let me know. Thank you.
Yeah, thank you so much, Komal. We would like to open the stage for anybody who would like to ask a question. Please feel free to raise your hand and we will bring you up the stage. Bob, you can check later if there are questions on the room chat. So I would just like to ask you, Komal, can you give us an example of networks with high hierarchy for the sake of those who just came in?
Network with hierarchy, you mean?
Yeah, with high hierarchy.
With high hierarchy, okay. Yeah, network with high hierarchy, a main example of this is actually like our blood veins. So, which actually, like not the microvasculature, but the main blood vasculature, which goes from our heart to the rest of our body, basically. There are these big, very thick veins which goes through our body, which are so as thick that we can actually see them. This goes to our rest of our body, when they're very close to the tissues in our body, then they create this very loopy, meshy network, and with very, very small tubes. So that means they have like a very strong hierarchy of what kind of tubes there are in the network, and there are very different conductances or different ranges of radiuses in this network, which is one same network, but it has a huge range of values of how thick these tubes are, which is what we consider as hierarchy of the tube diameters in one network, basically. And one example would be basically our blood vasculature, which has this very strong hierarchy.
Thank you. Can you also say the same for some plants?
Yeah, in case of some plants also, actually in case of plants also, this kind of hierarchy is observed, because if we look at a plant leaf, we can always see that there is this like main vein which is going through the center of the leaf, and then there are the small veins which are branching out from the main vein towards the edges of the leaves, and then they are also getting connected to then thin tubes at the edge, so there is also a hierarchy in plant veins as well. But this hierarchy is not as strong as the one that we observe in case of like in case of blood vasculature, because in case of plant veins, the way the veins are made, because in case of animals we have different way, the property of the veins are different because they are tubular and they are made with cells which surrounds which makes these tubes basically with hollow tubes, completely hollow tubes with fluid flowing through them, where in case of blood vasculature the tubes are different because there is like the links are different. Basically they are not like these hollow tubes, but they are like cells coming together and the fluid is basically passing through the cells, kind of. I'm not an expert in plant physiology myself, so maybe there is someone in the stage who can actually back me up if I'm saying something wrong. But as much as I understand that the tube structure is not a hollow cylindrical tube, and this is why the hierarchy in case of plant tubes are not as strong as in case of blood vasculature.
So I also want to ask, in terms of intuition that you have based upon the study that you made this time, like for example, you gave a specific case wherein some blockages in blood vessels in a case of stroke wherein there is coagulation. Even if you remove the clot on that part, it will not return to the particular channel that they had used before in order to promote the flow. So in what do you think this system that you have discovered, of course it's applicable to the human body and the vascular system, but as you know, of course I was just thinking about it because you're trying to study also complex systems. What are the other applications or ideation that you can get out of this as applied to other particular adaptive systems?
Yeah, thank you for the question. So basically, as you can see that the model that we are using in this particular piece of work is a very simple model. It doesn't really follow it doesn't very closely follows all the properties of blood vasculature. It doesn't very closely follow all the properties of plant vasculature. It's a very general model. And it's a very simple model and it's a very general model, and this particular model can be actually used to explain like an array of system. As example, this model can also be used to describe mechanical network as well. And mechanical network which are made with, like as example, springs and springs connecting with each other, and where if we consider that the spring constants in the mechanical network has some kind of plasticity and it changes to actually optimize the elastic energy of the spring networks, we can also use the same model to understand the property of these mechanical networks as well. And so in the recent days, there is a new trend which has come up, which is called neuromorphic computing, where scientists are trying to study how not necessarily neural networks which can perform and learn different kind of tasks, but also networks which are physical network. Like neural network is not a physical network, they're kind of a model of a neural network, but how even physical networks can perform and learn and perform different kind of tasks like mechanical networks or flow networks. If they can also can be used for computation, as example neural networks are used, can they also be used as computers, basically like physical networks? And this is because these networks, as you can see, they are very energy efficient, they're very robust, and they can solve an array of problems, especially if the stimulus is actually physical stimulus like flow or voltage in case of resistor network, or in case of mechanical network, some kind of stress or strain, which are physical stimulus. So you don't have to like transfer the information of the physical stimulus into some kind of binary numbers, which can be given to the computer to compute the problem. So this is a new trend which has come up, and the understanding of memory formation in flow networks or adaptive networks in general, which can explain the memory formation in mechanical networks, resistor networks, flow networks, all kind of physical, not all kind of physical networks, but an array of physical networks. They can be also useful for this particular research field. And along with that, we, as I said, that in our group, we are trying to understand the smart properties, or so-called smart properties or behaviors of this very, very simple, primitive organism, Physarum polycephalum, which is basically a single cell in shape of a network. And this particular organism shows an unique range of intelligent behaviors. They can solve problems, they can compute tasks, they can solve puzzles, they can solve an array of intelligent computational problems. And this organism does not have any neural networks, they do not have any organizing center. And it's very interesting to understand where might the problem solving ability are evolving from a very, very physical perspective, and understanding how memory can be formed, because memory is very, very essential for any kind of computational tasks, as you all know. How memory is forming in flow networks, in biological flow networks, this can also be helpful for us to understand the intelligent properties of this organism as well. Thank you.
Thank you so much, Komal. Yeah, I was thinking about neuromorphic computing also when I asked you that question, and I was thinking of other forms of complex systems, like even bacteria, and also during the early days of complexity when they were trying to introduce it, like having fluid dynamics as akin to the behavior of the crowd in disasters, for instance, how they flow. So I was thinking about that because maybe your study has a lot of uses in medicine, in computation, and in even just checking how to do something about disaster management, and even natural fluid dynamics. As you said earlier, there was also the mechanical flow of network of this spring on how they behave, and also in morphogenesis. So anyway, I would like to give the mic to Dr. Ulu, who is here with us. Thank you, Dr. Ulu, for joining the stage. Welcome.
Thank you. And thanks for a very interesting presentation. I actually had a question related to Cecile's. I do work creating network models from neuroimaging data like diffusion imaging and functional magnetic resonance imaging. And I was wondering if there are any assumptions that that your models take into account for the structure of the network if I were to apply some of your work to, let's say, the network models that we create from neuroimaging data.
Hi. Thank you for the question. No, actually, I have not thought about it, that if this model can also be used. I mean, I have thought about it, but so there is one assumption in that sense, which I would not know how, because I'm not an expert in the field, how we can transfer into understanding or into modeling neural networks, is how to consider the constraint of available material in case of neural networks. Because all the very simple models of neural networks that I am aware of, like Hopfield network, are not, they only consider that, okay, if a link is being used again and again, they're going to get stronger and stronger, which is also something that my model, or it's not my model, it's also a very well-established model of adaptation, since which is being used since 2000s or before that. This adaptation model is taking into account that the network is the links are gonna grow and grow if they're being used more and more. But the more interesting thing, which in my opinion, which actually generates the memory formation in this network, is basically the constraint of available material, that the tubes would have to shrink for allow for other tubes to grow, which is not the case in case of, as far as I understood, in case of typical neural network models. And I wouldn't know exactly how to use this approximation or this particular approximation of the available constraint of available material, how to translate that into a neural network like model. If you have some ideas.
Yeah, I was just going to say the analogy might be, so for example, I mean, when we're doing neuroimaging, we're looking at things at more of a macro scale, but brain regions that are considered to be functionally connected together sort of have similar patterns of activity. And those patterns of activity could be either stronger or weaker based on experience or based on exposure to specific stimuli. So I wonder if that's akin to, the tube shrinking or growing. Likewise, when we look at diffusion-weighted imaging, albeit it's a model of the white matter tracks that connect different brain regions that are related to the physical properties of myelinated axons. And so we've done some modeling related to that, looking at how, information flows throughout a network, and we can use the data that we derive from neuroimaging to actually accurately model how information flows in a brain network and how that differs in patients with different psychiatric conditions, and how they also respond to treatment to correct those deficits in information flow throughout the network. So there may be some biological sort of proxies to what you're talking about.
Yeah, right. That's actually very, very interesting idea. I have to think about it more. But yes, I mean, in a macro scale, maybe not in a microscopic scale, but in a macro scale, one can think about, I guess, like, as much as I know, like in case of associative memory or something like that, when a section of neuron is related to another neuron, if we can, there can be possibilities of transferring the similar kind of ideas, because this is what we are also using in our analysis as well, to track where this vanishing links are being created, which are essentially storing memory, and tracking what are the correlation to the stimulus basically. And similar ideas I think can be used, but I have to think about it. Actually, it's a very, very interesting perspective of also for the project as well. Thank you for bringing this up.
No problem. Thanks for the interesting talk.
Yeah, thank you, Dr. Ulu. I was just inviting Prakash there because we might have some questions about plants. So he's the plant expert. So Prakash, please feel free to come up. We might have some questions about plants. So anyway, here is Rajarshi, who came on stage, and also Nadje, who just dropped out. So I don't know if Rishi wants to ask a question or make a comment. Rishi.
I'm sorry I dropped in very late. I wonder if you wouldn't mind going over the constants in slides 11 and 12 and what they represent, if you wouldn't mind, please.
Hi. In slides 11 and 12, I discussed about how I am basically modeling the flow networks as a simple current network. Because when I'm talking about flow networks, flow networks can be modeled as a network with hollow cylindrical tubes joined with each other with fluid flowing through these cylindrical tubes, this straight hollow cylindrical tubes. And if I consider a straight hollow cylindrical tube with some approximations following a very well-established equation called Hagen-Poiseuille equation, one can write down the the fluid or the volumetric flow rate through these tubes, which I represent in this equation by Qij, through each of these tubes as linearly proportional to the pressure differences along the tubes. And the pressure differences, I represented them as Pi minus Pj along the tube. And if I consider them linearly proportional, I don't consider them, sorry. So this comes out from assuming Stokes' law and considering that the low Reynolds number fluid, linear flow through these straight hollow cylindrical tubes, and considering that this comes out automatically. And what comes out is that the proportionality constant, which is similar to a conductance in case of resistor network, and this is a conductance of flow, is basically related to the radius of the tubes and the length of the tubes and the viscosity of the tubes, which essentially means that if the radius of the tubes are higher, then the conductances of the tubes are also higher. That means more fluid is going to flow through the tubes if the radius of the tube is higher, even for the same amount of pressure. And similarly,
sorry to interrupt. And also in slide 26, you mentioned there's a difference between a hollow cylindrical tube and a porous tube. So are most of the transport networks like blood vessels and nerves better described as a porous tube or a hollow cylindrical tube?
So blood vessel networks are described well as a hollow cylindrical tube because here the epithelial cells basically come together and they make essentially hollow tubes for the fluid to pass through. But in case of plant vasculature system, although I'm not an expert of plant physiology, as much as I understand that they can be modeled as porous tubes because the cells, the fluid kind of passes through the cells in a blood vessel. The cells, because they have cell walls and they have very strong stiff cells, they don't really come together to form this hollow cylindrical tubes. They basically make space for the fluid to pass through the tubes. So each of the veins or the connections in the water vasculature in plant can be actually mapped something similar to a porous tube. Maybe not exactly, but kind of similar, more closer to porous tube. They can be mapped to. Yes.
Got it. So the reason I'm very, very interested in this presentation specifically because there's a few things in the realm of pseudoscience which I hold to be somewhat credible. For example, the common Eastern philosophy around chi networks or pranic energy. And I theorize that they could potentially be descriptions for the path of least resistance of flow for different compounds that are used by the human body. If it's water, there's like a path of least resistance for water. If it's oxygen, there's a path of least resistance for oxygen throughout all the different parts in the body. And I think potentially this might be a way to prove or disprove some of the theories behind acupuncture and these sort of Eastern medicine-based flow network theories, which don't have much credence in modern science. But I might have a few more questions, but thank you so much for taking them.
Thank you for your interest.
Yeah, thank you. I have invited Prakash here on stage because earlier I had the question about the veins of plants. So Komal here was wondering if there was somebody who can comment about plants. I mean, to probably confirm what she was talking about. So I don't know, Komal, if you have a question about plants to Prakash.
Hi, Prakash. Thanks for being here. So I'm a theoretician, and in my group, we are only working on blood veins of animals and on this Physarum slime or Physarum polycephalum on the network of them. So I'm not an expert of exactly how the plant veins are consisting of and what are their properties, because as much as I know, I only know more closely how blood veins are looking like and how Physarum are looking like, and I know that they are hollow cylindrical tubes, more or less. They can be mapped as, and I was wondering if plant veins have different properties. And as far as I know from literature, that they are they can be mapped to some kind of porous tube that the fluid is flowing through between the cells. Basically, the cells are not really coming together to make these tubes, but the fluids are flowing through the cells. And I wanted someone to give me some more insights on this. Thank you.
Yes, hello, Komal. Thanks, Cecile, for inviting such a wonderful speaker and holding this and pinging me into the room. A very interesting perspective, Komal, sharing this thing about memory formation. You know,
Prakash, come closer to the mic.
Oh, I still have. Is it better now?
Yes, it's better.
Okay. Sorry about that. Now, one of the ways, one of the most important intersections in biology is between what we call development. So there has to be signals for things to develop, and the development is very plastic. It does keep changing, especially in plants because they're always growing. And then you have signaling pathways, which allows signals to transduce across developmental spaces. And then you have the environment, obviously, right? The environment is feeding information. The reason I'm bringing this larger framework is you'll understand where information becomes structure, form, and function, right? So if you look at the development of a leaf vein and compare it to the development of a vascular system in animal things. In the animals, you have these kind of pericyte cells with seed, right? And then eventually you have a morphogenic field of information through which these pericytes become tubes. And they can be of different dimensions depending on all kinds of signals. So if you, and you can envision how these tubes form. But in plants, in animals, this program is mostly forward-looking, right? Mostly, that means that once they are differentiated, they just function and then remain there with a little bit of plasticity. It's much more in the vascular system than in other development in animals. In plants, it's very plastic. It's very, very plastic because they can form, and when they get injured, they can reform connections. The vascular connection is never lost. You cut a vascular system, it will reconnect. Same thing in animals, but not as much plastic as plants. And so if you look at that process of converting a cell into a cell that can conduct water is phenomenally regenerative. And I was thinking, when you were talking, because I really did not understand I had to go back and look at it because I just dropped into the room towards the end of the conversation. One of the things that I always wonder about is as this, think of, you're talking about flow and memory, and I was thinking about flow and information, right? And information flow in a morphogenic field can be connecting form and function. You know what I mean? So I'm going more fundamental, right? If you look at a plant, the plants are not really holding memory as animals, but they're always functioning, right? They're always connecting form and function. So they don't need a whole lot of high-level memory processing to happen because the memory is always there. They're always adaptive frameworks. So I was wondering if there is somebody who's working in your institute or other places, but they connect these two, the form, function, information, how it gives rise to these kind of stable living structures. Because I think that's where, we've talked about it earlier with Michael Levin's work of bioelectricity and how it connects, morphogenesis and information, right? In the same way, you're trying to connect a flow state of again, it's a flow state of information with reference to, for example, in a vascular system, amount of blood in a system, amount of oxygen in a system, for example. So I'm just wondering, going from a theoretical framework, are you linking up with some kind of a biology program or something to do with hard biology, which will allow you to test some of these? That's my second question. So, thank you.
Hi, thank you.
I think Prakash, if you will not speak, it's good to mute because we are having an echo. So Komal, please continue. Thank you.
Hi, no, thank you for the perspective. Yeah, so this is definitely a very interesting perspective. Like, so in our group, so what I can say from our group's perspective is that in our group we are focusing, we are using this particular very simple organism, which is very simple, and it's a single cellular organism, so it's very easy to grow and study and look at different kind of how do you say, morphological changes in this organism over time as a function of, we are mainly looking at fluid flow till now. But yes, in our group, we have also started looking into how because this is a single cellular organism, but it has many different nuclei. And in our group, we have started looking into how this nuclei and the activities of this nuclei, and how do you say, let's say of course the nuclei are also going to determine what genes are being actually transcribed at one point, and how the nuclei are correlated, or the nuclear divisions, and the location where the nuclei are, and at which point they are actually just flowing through the fluid, and at which point they are actually being used to transcribe some kind of some kind of protein to activate the network to do some kind of property. And this is also a new direction that we have started exploring, basically, and where we want to go into the connection of how the morphology, that is, I mean, in case of flow network, in case of Physarum, the morphology of the network, because the organism itself is generating the flow, which is not true in case of vasculature network, but there are other complexities in vasculature network. And the complexity of this simple organism is that the morphology itself is generating the flow, and the flow is feeding back to the adaptation of the morphology. What I presented here is basically the feedback of flow to the morphology, and again the morphology to the distribution of fluid flow in the network itself. And in our group, we have also started studying the correlation of the nuclei movement as example, and how that is changing, or that might change the activity of different parts of the network of this organism, and what actually generates the flow in the organism itself, and then connecting that back into the flow feedback into morphology. So there's like these different feedbacks, different sections of the feedbacks that we are trying to study from different perspectives. So I studied only one set of feedbacks, but there are different people in our group who are also working on studying different kind of feedbacks as well. I guess, I mean, this was something similar to, I guess I answered your question. I hope I could answer parts of your question, I guess. I don't remember what else you discussed.
Yes, thank you, Komal. That was very interesting, because you're talking about a kind of free, a nuclear space where they have multiple nuclei that are all distributed. You're looking for information structure or flow between nuclei where there's no boundaries. That's very interesting. That you have a very unique system. It's it's kind of between a plant-like system where the flow is diffusion and some kind of other pressure systems, osmotic pressures, with what controls flow in a vascular system, cardiovascular system, is quite different, because it's like a pipe. Whereas here you have an in-between framework where you have information and flow talking to each other. So it's very interesting. Thank you. And obviously the system you have, Physarum polycephalum, is a phenomenal system to understand how they, what kind of, within quotes, sentience, it's a catchword now, but what kind of, what kind of purpose, in biology you can't talk about purpose, but being a human being and having teleological vocabulary, I would say, what kind of drive does it have to, and it'll be interesting to see what kind of drive that morphogenic space reflects. It's very interesting interface. Yeah, thank you.
Yeah, it is actually really interesting organism to study all these different interactions coming together, but in a kind of seemingly simple system, which makes it kind of easier for us to study, because studying blood vessel formation, what we are also working on in our group as well, where we are making blood vessels on chip to look at how the blood flow actually changes the blood vessel development. And so this is what we are looking into, like into the development of blood vessel, but in not inside a body, but like on a chip. So that's already super difficult to organize, and where studying this system, it's much simpler, and it's just really unique in a lot of different sense. It's very unique. So yeah, it's very interesting, and thank you for all your perspective, and it was really interesting.
Osmotic pressure is something that we understand works at the cellular level. I think cardiovascular pressure also. What kind of other pressures might we look at when we're talking about, for example, human body, or specifically our work perhaps? Like it was modeled against voltage as a pressure, right, as an alternative to pressure. But what might be the actual physical mechanisms of? I mean, I'm trying to simplify it in my head to understand it, and from what I can understand, it's like the shape of the river bank is changed by the flow of the river, and therefore the river is able to flow faster or more efficiently through that space because it's shaped by flow over many periods of time. So, I mean, is that the physical mechanism, the change in morphology, or is there something else that I'm missing in terms of a pressure?
Yeah. Hi, Rajeshi. It's actually, you are absolutely correct. This is exactly the morphological change that I'm talking about here. That the network is basically like a riverbed, as you would think that there is like a. So the pressure here is basically. I'm talking about the pressure, not in this particular project. I'm not talking about the pressure along, how do you say, the cross section of the rivers. I'm talking about the pressure which is along the length of the river. In case of rivers, if I'm thinking about flow networks, in case of rivers, the pressures are generated by gravity. But in case of heart, or in case of our body, the pressure is actually generated by our heart, which is active. So in case of our body, that the pressure has to be like actively generated. The flow has to be actively generated. And as I said, like the organism that we are talking about, in case of Physarum, the pressure along the links are also generated by active contraction, active ectomyosin contraction of the cell of this, because it's a cell. Like the Physarum polycephalum is a single cell, so the pressure is generated by active ectomyosin contraction along the lengths of the tubes, which is called peristalsis. You can look it up. There are many different works which works on the process of how peristalsis generates flow. And so this is called peristaltic flow, where the contraction of the tube is basically making the fluid flow through the tubes, basically. And in the process of fluid flowing through the tubes, because these are adaptive systems, and as Prakash also mentioned, they're constantly changing and evolving. They also changes the morphology of the networks, and of course the morphology of the tubes as well, because in case of blood vessel, the cells around the tubes are constantly flowing as a function of the shear stress they're facing, which is coming from the fluid that is going through the cell, basically going through the tube. They're facing the shear stress, and then the cells are moving from one part of the network to another part of the network, making one tube bigger or smaller or stronger or less strong and stuff like that.
Very limited understanding of reinforces sort of neural pathways is that something to do with dendritic density and calcification of certain routes, so that it's easier for, I assume, potentials to pass through that direction. So how does that fit into this expansion of morphology in nervous systems? I mean, maybe I'm understanding it wrong what I said, but that's sort of how I remember it. If you could maybe shed some light there.
Yeah, hi. So basically, so it's the change of morphology as a function of the flow. It's kind of fits into it kind of has the similar idea of the very simple models of synaptic plasticity, like the Hebbian model, which basically suggests that a synaptic strength is going to, like in a very, very like crude way, let's say, not going into too much of the details because I cannot say I'm an expert of that either, that it suggests that in a very crude way that the synaptic strengths are going to increase if the synapses are being used more and more. And this idea is also kind of coming here in our work as well. But the interesting thing is that in this particular work, we did not implement this growth of tubes as a function of the strengths of the usable, like how much they're being used, and the growing of the connections as a function of how often they're being used or how much they're being used. We did not implement it like by ourselves. This is kind of like a phenomena that is emerging from the adaptation of the networks itself. Basically, this plasticity that is kind of being generated. It's emerging from the adaptation of the networks, and the adaptation of the networks is only trying to optimize the networks basically for efficiency, basically, and which is pretty common in all kind of biological systems, that the networks adapt to make themselves more optimized. And so this is what is this is like one of the main thing that the plasticity that we are seeing is kind of like an emergent phenomenon in that sense, kind of.
So if this is a over many generations, the gene pool is aware of this force that it's probably actively utilizing to achieve memory, either neurons or just more efficient pathways, right? Evolutionary over generations. So is there any evidence of blood vessels or neural pathways taking advantage of this in a more active way, like structurally? Like maybe the bronchioli of the lungs are designed to maximize volume to maximize surface area within the limited space, right? So the structures are a lot like a porous tube as much as possible. But the expansion of the areolas, I'm forgetting the word, sorry, is the expansion bronchioli. The expansion of the bronchioli probably takes advantage of do you see any evidence in blood vessels or nerve cells that the materials themselves were designed to expand in this way or take advantage of this phenomena?
In case of blood vessel, it's actually observed that the tubes, or like how do you say, the veins, which has high amount of blood flow, they actually expand over time. And this is the biological phenomena that it has been observed, or the other way around, that the tubes that are not being used over time, they basically shrink over time. And then this adaptation is basically like the biological mechanism behind it is basically the epithelial cells that are creating the blood vessels, so they can sense the shear stress of the flow, the blood flow, and they kind of follow the shear stress of the blood flow to find a location where they can find the optimal amount of shear stress that they're comfortable with. So basically, they kind of detach from the blood vessel and flow along the blood to gather around a different blood vessel to make that blood vessel thicker and stronger, and basically prunes away the blood vessel where they have low shear stress, or some like they basically adapt along with the shear stress of the flow, and they change the adaptation, and therefore ultimately what comes out is basically that the tubes glow when they have high amount of flow through it, and tubes end up shrinking when they have low amount of flow through it, because the cells are basically moving around as a function of the flow, or after sensing the flow through the tubes.
Oh, got it. Thank you. That's very interesting. Just one last question for me. So, if how do I put this? So, what is the biological mechanism for detecting these stresses? Like, how is it? Is it accumulation of some chemical compound that acts an indicator for how much stress over time, or is it how does the body keep track of how much stress over time in a certain space?
I am not sure that in case of blood vessels, the exact biological mechanism of how they are sensing the stress is known or not, but it is in general well known that cells can sense mechanical properties. They can sense the surface in which they are glowing, they can sense the amount of force on them, or they can sense mechanical properties, and then they change, the cells basically change their own mechanical properties, because as you said, like, I am not 100% sure of the exact biochemical pathway behind it, but yes, like some kind of external mechanical forces can actually how do you say, can trigger some pathways, some chemical pathways that basically changes that starts some kind of chemical reaction of the actomyosin cortex of the cell, which changes the mechanical property of the outside of the cell. So like if it is growing on a stiff surface like bones, basically, like cells will become into bones, and if they're glowing on a very soft surface, then they will become into some other cells with different mechanical properties of the cell wall, basically, or like the cell itself. Like, and I'm pretty sure that there are like many different pathways which are coming together to generate this mechanosensing properties of cell. And there is a huge community who are working on different perspectives of and different applications of this property of cells, actually, on this mechanosensing properties of the cells. And there's like a huge community who is working on this topic. This is super interesting. They're like growing how do you say, like the cells in the like the bone marrow cells into different kind of surfaces to create different kind of cells, like heart cells or bone cells and different kind of things. And there's like a huge community working on this, and there are like many different pathways which are coming together, as much as I understand. But I would have to look up into figuring out what exactly is the current knowledge of the scientific society into this.
Thank you so much for taking all my questions. That's a very amazing presentation.
Yeah, Komal. I promised you that at 1.5 hours, I will be asking you how much more time would you have for us. So we are like 1 hour and 45 minutes actually here. So I want to ask you if you have a little more time for us here.
Hi, Cecil. Yeah, I have more time, and if there's any other questions or comments or anything, I would love to talk. Yeah.
So thank you. Your study is really very interesting, and I think it will lead to other studies as well. So yeah, I would just like to ask you, taking it from here, I know that your lab is making other experiments which are related to this, and but anyway, based upon the paper, your paper that you discussed now, what are your next steps?
So as my next step, like as a follow-up on the work that I presented here, what we started studying is basically theoretically if we can quantify what is the memory capacity of such networks. Because till now here we talked about how memory is being formed in such network. We probed for one stimulus information, but the next obvious question is, can this kind of network remember multiple stimulus? Can we overwrite the information of one stimulus written into this network morphology by applying another stimulus? And like the obvious question that comes up whenever I think the concept of memory is discussed in case of different models of neural network or different models of memory systems, that what is the capacity? And this is like the next question that we actually have a archive like a publication which is on archive on this topic. Maybe I can share the link with Cecile and she can share it here. And also along with that, we are also trying to figure out if we can, so we also have, I think I shared a paper link in the work in the presentation here as well, that we studied the phenomena of memory formation in this organism as well, this simple non-neuronal organism, single cellular organism, and how memory is being formed in the network morphology. And this is something that's also published work in PNAS from 2021. And now we are also trying to understand if we can understand memory capacity of such organism or not, as well from an experimental perspective.
Yeah, thank you. Yeah, so many implications of your work. I cannot. There is so much to imagine and ideate on what it could lead to, and also its use cases. So yeah, we maybe Bob, there is a comment by Oberlunar. I think Oberlunar here on the room chat that you may read in a while. And I think Prakash even also tried to recommend a book about plants, about the veins of plants. And so, yeah, if you'd like to read that. In the meantime, there is Rahul who came on stage. I wonder if Rahul has a question. After Rahul's question, maybe Bob, you can read the comments in the room chat. Thank you. So, Rahul, would you like to speak? Yeah, if you can speak now, maybe Bob can go to the chat. Bob?
Can do.
Thank you.
So this is a question, well, first a comment and then a question from Oberlunar. Thank you for your presentation. The computational smart capacity of tube geometric adapted networks with respect to, for example, spiked neural networks or typical backprop-based neural models would be an interesting comparison.
Yeah, that sounds interesting, but I never actually looked into it. But yeah, sounds like a very interesting itself. I know that there are also papers who studied how flow networks can learn using inspiration from the neural network models, as example, using the backpropagation methods of neural network adaptation to make these flow networks learn. Yeah, that would be something interesting. If that's a perspective that we have also started thinking about, that how to evolve the model into making it more robust and maybe we would need to use some other adaptation techniques or evolve it to make it more robust into learning more things, let's say. But yeah, that's very interesting perspective. Yes, indeed.
Thank you.
So Komal, this is a little bit funny. I was just thinking if these networks are forming some kind of memory, and of course they have their memory because they learned something. And I was just thinking, in case of possible use cases, maybe in the future, if they remember something because they have memory, is it possible for them to unlearn what they had learned, so that that particular network can be used again? Because you see, they atrophy, right? If they are not used anymore, they have a weak link, and then finally in the illustration that you showed, if they are no longer used, so they disappear. So I was thinking, if on the process of not disappearing yet, since they are learning something from memory, maybe for some applications like in medicine or other applications later on, if I'm just curious. I know that you are still studying it, but what they had learned can do have some possible use to that network, which was going to cease to have use. So yeah, I was just thinking along that line.
Yes, Cecile, thank you for this question. It's actually very interesting. Actually, this is a perspective that we tried to answer in our archive paper that I might have sent you a link to. So there we were studying basically the capacity of networks, the capacity of these networks, the model that I'm used here, that's not my model, but it's a well-established model, and what is the memory capacity of such network. And what we in fact see is that if we continue using this particular model, which is a very, very simple model, I have to accept that it's a very simple model. What we see is that these networks actually cannot unlearn. They basically get old. Like if I write more and more information over a long time, this networks just gets saturated with information, and they cannot take in more information anymore after a certain amount of time, and after a certain amount of information it has already learned. But yes, that is a very, very interesting perspective, because always whenever that idea of memory comes in, we always have to think about forgetting and how to implement forgetting, because forgetting is very, very important for a network or for a system to robustly learn many different things to increase the capacity. So I guess, there are different ideas one can think about how these networks can unlearn, and one would be that in what happens in case of Physarum network. If I'm thinking again, it's a simple system, so I'm always thinking from the Physarum network perspective. What these networks do, basically, they move away. They don't stay at one place. So they shrink tubes, and then they create these strong hierarchies. They keep some information in the hierarchies, but after a certain amount of time, they move away and they create a whole new network, kind of a way of erasing all the information they had before, and a way that creates or that allows them to learn more information because they're moving away from the space itself. And this is a self-sufficient organism, right? It's not like part of a whole body like our blood vasculature, which is a part of our whole body, and it's a different situation. But in case of Physarum, it's a self-sufficient organism. It's just like surviving on its own, and they do this. So this is like one mechanism that could allow the network to unlearn, which is basically growing totally new tubes, basically totally new connections, and erasing whatever they have over time. That is like the biological thing, because activity is also going to like eat away everything, and creating totally new connections, and as they're moving along. And this would be like one mechanism what I would say that they can unlearn. And also, as Prakash also mentioned, that plants, as example, they're constantly growing, and they're constantly growing new links, new things, new cells, and they're like they have a lot of plasticity going on. They have a lot of activity going on, what exactly Prakash said. And this would be another method of like another way the networks could just erase information from the network morphology, because they're changing the morphology completely after a certain amount of time, which happens in our body as well. There is like the cells are going to move away completely after a certain amount of time. So there are definitely in biological system, in real systems, there are implementation of different models one could use to basically introduce the forgetting abilities of such flow networks as well. Yeah, thank you.
Yeah, thank you so much. Yeah, there's so much to imagine, and so much to really try to ideate on the use cases of something that you stimulated that, as you say, it's just very simple, but actually it has a lot of it will, I think, contribute to many understandings or what we still do not know about science in different fields. So actually, I was just reminded, I was inviting this researcher who did an essay. He did a paper, a journal article about Gabriel Garcia Marquez in 100 Years of Solitude. So he was making this essay about the importance of forgetting and remembering and creating networks of memory. And yeah, it just so happened that you had this talk. It's just an essay actually, but it's done by the Department of Neuroscience of by a neuroscientist in the publication of Oxford Academic Brain. And I think this is going to be a good storytelling to people who would like to understand science more. And now I'm thinking about its relationship in the study that you do. So thank you so much. You have inspired us, with the Physarum inspiration. I think I will look more into Physarum now and maybe treat it with more respect.
Thank you. That's also very interesting paper article that you mentioned. I will also look into it. I also saw some other memory and forgetting relationship articles as well. It's a very interesting perspective to think about. Definitely.
Yeah, thank you, Komal. We have Hatam, who came here on stage. Hatam, hi, Hatam. I'm sorry if I mispronounced your name.
Haitham. Is your name Cecile? Hello?
Yes, it's Cecile. Sorry.
No problem. I just want to understand the topic of the room right now, because I've just arrived. Memory formation in adaptive networks. What do you mean by adaptive network? You mean when you align the things that you want to remember to other things, or you connect them to other things like mind maps and so on and so forth, or the so-called memory palaces. You mean this?
Hi, Sam. Sorry if I pronounced your name wrong. So by adaptive network here, thanks for your question, by the way. So by adaptive network here, I meant to talk about basically physical networks, which that is like my main focus of my PhD and my work, which are mainly physical networks like mechanical network or flow networks, which change over time, which adapts by itself. There is not no one is intentionally changing the network connection and the network connectivity. The network is adapting, growing to whatever conditions that the network has, environmental condition or internal conditions. It is adapting itself. This is in a very, very general perspective, let's say like that. So this is the adaptive network, the kind of adaptive network I thought of. And if you want to compare with, in my opinion, one kind of network, these physical adaptive networks are similar to our social network, where the connection between people, the links between people, the links between us and our friends, or us and our peers and family is adapting constantly depending on our interactions, depending on our life and different situations. The links are constantly adapting, and this is the kind of adaptive network I meant to say, which is like adapting by itself, where the link connections are adapting by itself. But the main focus of our work is actually adaptive flow networks. But the work can also be implemented to adaptive mechanical networks or adaptive resistor network, let's say specifically physical networks as well. But the main focus of our work is actually adaptive flow networks, like our blood vessels, which are changing by itself constantly depending on the blood that is flowing through the network. I hope this answers your question.
I just want to say that I had posted on the room chat the paper of Komal about the memory capacity of adaptive flow networks. I think if you read the PDF of that, if you have time later, it would be a good reference. So, yeah, we have Armish here on stage. Hi, Armish. Welcome.
Thank you, Cecil. Thank you, Komal, for your presentation. My question is as follows. In the adaptive network which you talked about, how much of a change is observable or measurable over a period of time? So can we measure the changes in the lab into these networks? That's part A. And if we can measure those changes in the network structure or size, can we generalize it? And the second part is, if let's say we get a pattern, because we all grow and into certain age, I think most of us are similar biologically. So can there be a generalization of these networks?
Hi, Ramesh. Thanks for your question. So there, like as I said, as we already talked about with Prakash as well, that adaptive flow networks can be very different networks in different organisms. They have different time scales over which they are changing at different phase of the development. As example, an organism which is in the developmental phase, like when they are like, let's say, like fetus or something like that, like and then in the developmental phase, there is definitely a lot more adaptation of the network which is happening. The network, like when the organism is growing, basically there is a lot of adaptation happening at that time, at the growing phase. There are some things like the seeds, like from where the networks will start growing from. There are some things which are encoded in our gene, but afterwards the network actually grows depending on whatever random variable it comes across in the organism body. I'm thinking of blood vessels here, and yes, you can actually during the developmental phase, especially, you can definitely very well track. Like there are lots of researches on tracking the adaptation of blood vessels networks. In case of zebrafish, which is a very interesting model organism to a lot of scientists studying blood vessel adaptation, because they have like a very simple body which allows people to like really look through how the blood vessels are adapting over time when they have like a certain gene is actually like shut down or like inactivated in the organism, when they're medicated or when they're injected with something else. Like they can very well see how the blood vessels are changing over time, and they can like really, they have documented different relationships. There's like also like a huge group of scientists which are working on that, and so there are like there are a lot of studies who are working, and as I'm at the top of my mind, I can think about the zebrafish studies, because there are a lot of them, because these are very very nice organisms to look at blood vessel adaptation. And then because they're kind of like see-through and they're like young. And then there are also blood vessel adaptation studies in case of chicken embryo as well. Chicken embryo, when they are looking at how important it is in case of embryos to, in case of the blood vessels in embryos to actually this adapt and mold. Like it's super duper important. If they cannot do this, these embryos actually die if they don't have the properties, the abilities to adapt. And they have like observed and noted down and how the blood vessels are actually changing over time. And I think the part 2 of your question was, are there general mechanisms or general, if the dynamics can be generalized or not? And that I mean, of course they can be generalized to a certain scale. It depends on like at what properties you are looking into. If you go into too much details, they cannot be generalized after a certain point. But like the things that I based my ideas on, or not my ideas, but the model which I used, the idea that they are using basically is that the tubes, like in general, the blood vessels, the cells can sense the shear stress, the mechanical shear stress coming from the blood flow through these tubes, and they are adapting to actually accommodate for the flow through the tubes. And this is something that they have seen in general across organisms. And this is something that can be generalized, and they have found. But there are definitely like a lot of research still going on figuring out what exactly is changing with different parameters and across different organisms. And they're still, of course, it's an ongoing study. But yeah, there are some studies which have shown this. I hope I answered your question.
Thank you, Komal. I just have a quick follow-up. Does this adaptation always happens in response to stimuli, or organisms have developed some sort of muscle memory which also works in parallel, like without any stimulus it grows in a certain fashion, which basically could have been passed on from generation to generation over a gene or some sort of mechanism. So the question is it just first stimulus happen and then the change in structure happens, or the change is inevitable to a certain extent because that's the muscle memory that organisms have learned?
Yeah, so the adaptation itself, as much as I understood from your question, if the adaptation happens only in presence of stimulus, so the adaptation actually happens all the time. Like it's just the way the tubes, the networks react no matter what circumstances they're in. Now the circumstances are kind of the stimulus. As example, because there is a lot of randomness in the environment that like the organisms are growing in, and this will come into play during their development. Basically, I mean, I'm not a doctor, or I'm not an expert in this field, that I cannot say very strongly that how much effect the development of a baby can have depending on the habits or the physiology of the mother. And I cannot say very strongly, but like if I'm thinking from my naive perspective, then I would think that yes, I mean, like as example, like the randomness of the physiology of the mother is gonna change the stimulus, let's say, or the circumstances or the flows, and the blood flow and the blood pressures that we talked about, and the amount of blood pressure that the fetus is going to have, and all these things, they're going to change. That's going to change that, and that's going to definitely change the way the networks adapt. this is as much as I know, but I don't want to comment very strongly on this because I'm not an expert. But very vaguely, yes. I mean that they adapt all the time, and they adapt as a reaction to different circumstantial pressures and like the properties of the cells itself, which can be determined by the genes, and some genetic structures that determine the initial positions of the blood vessels or how the blood vessels are going to start growing. But then the angiogenesis and everything is going to be dependent on different randomnesses, let's say, of biology, basically. I hope I answered it.
Yeah, thank you. This is very helpful. Thank you.
If I could interject something, we've gone over our allotted time, and I don't want to abuse our wonderful speaker, Komal. So can I give the microphone to Cecile to do our wrap-up? That would be great. Well, perhaps. Okay.
Yeah, thank you so much, Komal. All your discussions here, I also would like to thank the people who participated in the discussions. I think we can just go on and on, because I remember seeing a recent study also in neuroscience on I was just thinking, what are the driving forces that probably would be able to move adaptive networks as regards not only to the stimuli that is being given to them, but that the course of action that they are taking, because of course you're talking about the vascular network here, but on the other hand, I just remember a certain study that appeared talking about on how we absorb information and on the plasticity of the brain, wherein it's not just only the neural networks that are also moving, but to keep the information, but like there is something that outside, there are some substances that they're trying to study that tries to pin your memory at the age 7, because as you see, the child is like a sponge, and even if you teach languages, they are very prolific about absorbing it, but it has to be pinned, or else if there's so much information, people will forget that as well. And then there is also a certain phase wherein we are trying to remember something after like 7 years old, and this is a new study that they had just made, because this is different from the previous ones that we thought we understood. So, but maybe we can discuss it at another time when you have more studies and we have more time, because I know that you have a limited time to stay here also, so we don't want to take up most of your time. So yeah, I just would like to thank you. We wish that we can have maybe an open discussion in the future, and I would like to look at the other papers that you have. Probably we can make another room related to it, because it relates to other fields of discipline. And anyway, Komal, it was very, very interesting and a very succinct presentation, and also very great engagement with the audience. We would like to prepare to wrap up, and thank you so much really for having this incredible study, which has a lot of implications. So yeah, I would like to ask you if you have some message to the audience as we try to wrap up.
Thanks, Cecile. Thanks for organizing, and thanks for having me here. This was really really interesting and enlightening to think about all the perspectives that I even didn't think about, all the different ways like all the different perspectives of this project. So this was really interesting to talk to everyone, and I really thank everyone for listening to me and bearing with the presentation, and also staying so late listening to our discussion, and also asking for, and all the questions, other people who asked their questions as well. These were really, really interesting. Yeah, thanks again for having me, and I hope you enjoyed the talk. And if you have any questions or any other comments or anything that you would like to talk or discuss or anything, then you can always write me in Clubhouse, and I will send you my email ID to have longer discussions if you have any other questions or any books or any suggestions, anything that you want to comment on. Thank you again.
Thank you, Komal. I think we will invite you back soon. This is a good way to do some kind of creative thinking here with the science peg. So, yeah, I would just like to announce the upcoming rooms that we will be having. So, the two upcoming rooms that we will be having is about the impact of lack of sleep on the brain and the cognitive development of children, which will be done by Dr. Ze Wang of the University of Maryland. And after that, there will be another room on shaped single photons on integrated diamond nanophotonics. And our lecturer will be from Harvard. So yeah, please follow Quantum Photonics, and please try to look out for really good rooms coming up. So with that, since Darcel is not here, I would like to give the mic to Bob to help us on the countdown to close the room. Bob?
There I am. I was on the back channel. Sorry about that. Personally, Komal, I wanted to thank you because you've caused me to think differently about memory, and what a wonderful thing that is. So, since we're running out of time, we're going to go with a 5 count. And if everybody wants to unmute and join in, we're going to go with 5, 4, 3, 2, and Komal, you are number 1 for us today. Thank you so much.
1.
Yeah, thank you. Thank you, Komal. See you back in Quantum Photonics soon. And thank you, everyone who had listened to us. And see you in the next rooms of Quantum Photonics. Yeah, bye for now.
Bye.